Probably you feel tired, but let me start my talk. Okay. These days, I always start this kind of slide. My science is a system of knowledge accumulated by humanity as a whole, and science is a social activity, so we cannot be independent of the era we live. So today I will start my history. I was born in 1945. That's just half years before the World War II was over. At that time, everybody in Japan is so poor, and we have so many trouble about their food, and I had a hard time. My mother suffered from TB for a long time, and when I was in this high school, I was interested in chemistry, so I want to be a chemist when I entered the University of Tokyo, but as you know, 1960s was a really good time for just establishing the molecular biology. So I interested in the molecular biology pretty much. So I started in the lab of Kazutomo Imahori's lab, working on the ribosome, protein ribosome structure and the protein synthesis initiation mechanism. So my career, I'm pretty much interested in the protein synthesis within the cell. And as you know, biology needs just organisms to work. I have been already working on this small, tiny, single cell organism, Saccharomyces cerevisiae. It's just 5 nanometer long. It's a quite small organism, but it's very important for a human because we're getting many products from Saccharomyces cerevisiae. And this organism is also quite a good system for to understand our cell, so-called eukaryotic cell. So whole genome sequence, and we have so huge database on this organism. When I come back from the United States, from Rockefeller University, my boss said, you can work on any subject. So I wondered what is the quite interesting thing for me. And I'm not a competitive person, so I do not want work on a familiar, popular field. So I interested in just this quite simple organelle vacuole. When at that time I was in a botanical department. And so I interested in the vacuole because at that time, many people thought just vacuole is just a garbage trash in the cell. But why plant cell has so large vacuole? All of you probably know from the elementary school or middle school or high school, you learn plant cell has a large vacuole, but you probably, not so many people think about why plant cell has so big vacuole. So I want to say it is a big difference, and to understand it's a very big difference. And if we have time, I won't discuss about this later. So I think there are many, many things to be answered in nature. So about 10 years, I worked on a vacuole membrane, and I found vacuole is not inert organelle. It takes lots of amino acid or calcium or many kind of materials are stored in this organelle. And the driving force is a gradient of proton. Proton gradient is made by new type of proton ATPase, so-called V-type ATPase. Then in 1988, I got a small lab, I started by myself at the Tokyo University. And at that time, I want to change my subject to another new subject. And I want to understand the lytic function of yeast vacuole because, as I said, vacuole is an acidic compartment via V-type ATPase. And it was known vacuole contains various kinds of hydrolytic enzymes, protein degradation, nucleotidase, monosidase, phosphatase. So that must be the lytic compartment, degradation compartment. So I thought that might be homologous to the lysosome, so-called lysosome in mammals. I don't need it. So as you heard from the first talk today, all proteins are made by the genetic code in DNA to RNA to protein. And these 2 decades, we learned all proteins are made in the cytoplasm, and they are transported to the proper compartment or many kind of their organelle. So we learned how protein localized properly. But this just figure, we don't know about the fate of each protein. Each protein has a quite distinct lifetime from few minutes to 3 months or more. So before I go to the, my work, our work, I want to say several general scheme of the protein degradation in our body. And when I was I had a biology class at Tokyo University. Always I started my lecture from this question: how many blood cells are made per only 1 second in our body? You can easily calculate within 2 minutes. Okay. The answer is red blood cell is made 3 million cells per second. And how about hemoglobin? Hemoglobin is within red blood cell, it's bound to oxygen. And this kind of molecule, 1 times 10 to the 15th molecule per second we are making. That exactly means this number of cells and proteins are degraded. Wow. And okay, construction seems more positive, and disruption is a negative way. But in ours, for example, this is Tokyo University campus. In the spring, you can see leaves develop, and in summer, you can see lots of green leaves and actively photosynthesize. And in autumn, you can enjoy these yellow colored leaves. You know these kind of phenomena every year. But here, why these leaves turn yellow? That means all the chloroplast, the green pigment, photosynthetic machinery is degraded, and all the amino acid transported to the trunk. And another example is this is a rice field. In the summer, you can see green leaves, and by sunshine, these leaves actively photosynthesize to starch. And in the autumn, you can see yellow leaves. Again, exactly the same thing happened here. The reason leaves turn yellow, that means that all the protein in the leaves are degraded completely, and all the amino acids go to this grain of the rice. So for rice, degradation is very important to make next generation these grains. So actually, degradation is not negative way. It's very important. And another example, we are making every day about 200 to 300 grams of protein, but we are taking 70 to 80 grams of protein as a food. And if we may hold these amino acid, the protein turn to the amino acid, it's simply 70 grams is not sufficient for make this kind of protein. So just these simple numbers suggest our protein in our body degrade, and most of the amino acids come from our protein, our own protein. So our system is just the equilibrium state of the protein synthesis and degradation. So actually, life is an equilibrium state between synthesis and degradation of protein. So to my supplies, almost every 3 months, almost all protein in our body is replaced. So recycling is really essential for life. The first breakthrough in the intracellular protein degradation was made by Christian Dubé, Belgian cell biologist. He found a small membrane organelle called lysosome that contains lytic enzymes within. And then Rockefeller group, including Christian Dubé's group, analyzed how protein get into the lysosome. And they found, they named their own cytosolic protein transfer to the vacuole, to the lysosome. That means a self-eating process, autophagy. This is he coined the name of the process as autophagy. Since then, another very important protein degradation system was found, that is our ubiquitin proteasome system. That is very important to degrade a specific protein, just tagging the small protein, ubiquitin, and degrade by huge proteasome. And this is very important for various kinds of biological system regulation. But lysosome or vacuole system, almost nothing was known about the molecular mechanism of genes involved in that process. As I said, I was working on long time on the vacuole, yeast vacuole, and I want to know is really the lytic compartment within cell, because this is acidic compartment and contains very lots of degrading enzyme. So the questions I had is when and what and how cytosolic protein can across this vacuolar membrane and degrade it. First, I thought, what is the best condition to work with? Yeast cell, if you have a sufficient nutrient, yeast cell can grow just this kind of budding. But if nitrogen source, amino acid, the source of the amino acid, nitrogen starvation, induce this kind of quite dramatic change of the cell. Meiosis occurs, and these diploid cell forms for spores. This process is very important for genetic analysis of the cell. So this quite big change within cell should be done without any supply of the nutrient outside. So this process should be required lots of own protein degradation. So I thought nitrogen starvation might be very good condition to study the protein degradation. And I thought if I see the early step of the sporulation under microscope, I might see something under microscope, but not. Unfortunately, I couldn't see anything under light microscope. One day I thought, if I use the proteinase deficient mutant cell that was already constructed by Elizabeth Jones in the United States, I got these kind of the triple vacuolar proteinase deficient mutant cell. And if I use this cell under starvation condition, if I'm lucky, the structure or something delivered to the vacuole to be degraded might be seen under light microscope. Okay, this is a phenomena I found. Under growing cell or just the zero time, yeast has a vacuole, but nothing inside in the vacuole. But after 3 hours under starvation condition, you can see these kind of spherical structure are moving around in the cytosol. And to my surprise, almost all cells has this kind of structure. So this might be quite interesting phenomena. And so this was a starting point of the whole work. Still in my lab, many people just observing this phenomena. So this is a starting point, and always we are asking why this kind of phenomena happen under starvation conditions. This is the time course of the phenomena I showed. After 30 minutes, we can see this kind of small structure appear in the vacuole and gradually increase. And after 3 hours, almost all vacuole occupy this kind of structure. So we started the electron microscopic analysis of this phenomena. Mrs. Babar, my good collaborator, worked on this subject. And this is a 3 hours starvation starved cell. You can see within vacuole this kind of structure. Even this small vacuole contains. And this high magnification image, you can see the structure is single membrane bound. And you can see these black dots are ribosome. And you can easily think about the inside of this structure is really a portion of cytoplasm here. So from this picture, we thought nitrogen starvation induced this kind of structure for delivering cytoplasmic component randomly, non-selectively to the vacuole. You can see various kind of cytosolic structure within this structure. We named this as an autophagic body later. Here you can see a Raphael here. And many electron microscopic studies, we found this is a vacuole. Next to the vacuole, you can see this kind of cup-shaped membrane sac appear, just enwrapping a portion of cytosol. And this is a already enclosed double membrane structure, autophagosome in the yeast. And these double membrane structure, outer membrane of double membrane structure is continuous to the vacuolar membrane, and the inner membrane is just showing here. And these are the inner membrane structure released in the vacuolar sap. We so-called autophagic bodies. So, and as you see here, here's just forming a double membrane structure here. You can see this structure contains ribosome. Ribosome is already quite quite big structure, but you can see mitochondria here and here and here. That means the autophagy is quite unique system, degrade the whole organelle by enwrapping a portion of cytosol. So it is quite different strategy if we compare the ubiquitin proteasome system that strictly recognize one protein that should be degraded by sophisticated enzyme system. But autophagy is quite bulk is good for bulk degradation of own cytosolic component in the lytic compartment. So almost 30 years ago, we drew this kind of picture as a autophagic process in yeast. When yeast cell face various kind of the nutrient starvation, suddenly this kind of small membrane sac appear and elongate and form a double membrane structure, autophagosome, and autophagosome target to the vacuole, and the fusion event occurs, and the inner membrane structure are released in the vacuolar sap. But this structure is so quickly disintegrated in wild type cell within half minutes or so. So nobody found this kind of structure. I used the vacuole proteinase deficient mutant. I could follow the autophagic process as accumulation of autophagic bodies within vacuole. So next, we tried, obviously, we tried to analyze the mechanism of the autophagy in yeast by genetic approach to make, by isolating the autophagy defective mutant. If we could get the autophagy defective mutant, we can easily get the genes required for the autophagy in yeast. But we didn't know anything about what is the phenotype of the autophagy defective mutant in yeast. So we started the microscopic screening. No autophagic bodies accumulation within vacuole, just under microscope one by one. And my first graduate student succeeded to isolate only one autophagy defective mutant. At that time, we called APG1. Now we named ATG1 mutant. But vacuole is a quite complex membrane phenomena, so we should have more ATG genes. But ATG1 mutant is quite normal under nutrient-rich condition, standard yeast medium. But we found ATG1 mutant cannot survive for a long time nitrogen starvation. After 2 days, ATG mutant start to die. So I assume this loss of viability phenotype is caused by the defect in autophagy. So we tried the loss of viability phenotype as the first screening, and then we again use a light microscopic screening the mutant lack the autophagic body within vacuole. And this way we could easily get about 100 autophagy defective mutant. And just a genetic analysis, we could get 14 ATG genes as the first screening. Then now we say 18 ATG genes are essential for their autophagosome formation. So the first screening was quite efficient. That helped a lot for further analysis. And next question, what is the ATG? What kind of protein is coded by ATG genes? We started the cloning of ATG genes, sequencing of ATG genes, and identification of ATG proteins. But first 3 or 5 ATG genes, all were novel genes. We couldn't say anything about the function of ATG genes from that kind of analysis. So it was really a hard time for us. But I have no time to go in detail. But first we found quite interestingly ubiquitin, autophagy requires 2 kinds of ubiquitin-like conjugation system, ATG12 is much bigger than ubiquitin, but it's activated by an enzyme to transfer to another protein by this kind of thioester intermediate, and finally form a ubiquitin-like isopeptide bond formation with ATG5. And more interestingly, we found another ubiquitin-like protein within our genes. ATG8 is and C-terminal arginine is removed by this proteinase, and glycine, C-terminal glycine is activated again, the same E1 enzyme transferred to ATG3, and to our supplies, ATG8 is not conjugate with a protein, but it's a membrane phospholipid. Phosphatidylate amine was the target of ATG. And within several years in the National Institute of Basic Biology, we could find many new findings about ATG genes. And initiation is toll kinase is a very important upstream regulator of the autophagy, because if we add the rapamycin, they inhibit TOC1 even in quite nutrient-rich condition, autophagy is started. And I will show you a little bit about this initial step of autophagosome formation later. And within several years, 18 ATG genes were classified into this kind of reaction subgroups. ATG1 is a protein kinase, and ATG13 is an important regulator of the initial step of the autophagy. And we found PI3 kinase. It's a membrane phospholipid, inositol phosphate is phosphorylated by this kinase. And PI3P is generation is very important for the membrane dynamics. And as I said, these two ubiquitin-like conjugation is required for the autophagy. And ATG9 is the only membrane multi-membrane spanning protein among ATG genes. And yet we cannot say exactly the function of this complex, ATG2 and ATG18 complex. So ATG genes are just required for the autophagosome formation we found. And when I was lucky in NIBB because Tamotsu Yoshinori and Noboru Mizushima, they joined our lab and they started the ATG genes in mammals, and two postdoc and graduate students started to work on the plant ATG gene. And we found ATG genes are well conserved from yeast to human and plant. So identification of ATG genes was completely changed the landscape of autophagy research. The reason is, before, as I said, autophagy is only detectable by using electron microscopy. But now, Noboru Mizushima made GFPLC3. It's a homologue of ATG8 in yeast. and tagged GFP and expressed whole body transgenic mouse. If we analyze this mouse, we can say where and how much autophagosome is formed just under fluorescence microscope. This is a muscle, and this is a liver cell, and this is the fertilized egg. So we don't need the electron microscopy anymore. And as you see, this is a fibroblast. Under starvation, you can see lots of autophagosomes is formed. So this is actually degradation via autophagy. So now we know autophagy has a major two laws. One is, of course, nutrient recycling. That's very important for survival mechanism. And another important aspect of the autophagy is elimination of excessive or harmful materials from cytoplasm. So these are quite many people interested in this selective degradation of harmful mitochondria, lysosome, sometimes CEL, and a portion of nucleus, and also invasive bacteria or virus particle via autophagy. So, but we are, our group interested in how ATG gene function for autophagosome formation. And we found this is now we fluorescence microscope technique was very developed, so we can see ATG17, how that behaved in the cytosol. Within cytosol ATG17, it's actually a dimer of ATG17, 2931, just moving around in the cytosol, and under starvation condition, a portion of ATG17 stick to this kind of dot structure we named pre-autophagosome structure. This is the site of the autophagosome formation. So we think under, for example, nitrogen starvation, ATG1, ATG13, 2931 complex is formed next to the vacuole that we call past, and then ATG9 vesicle come and PI3 kinase join to the past, and ATG218 complex is joined. And finally, 2 conjugation system is required for the elongation step of the isolation membrane. But just we showed this, every protein is just a circle. But now we, by collaborating with Dr. Inagaki and Noda's group, we determined the 3-dimensional structure of most ATG protein. That helped a lot to understand the molecular detail of the autophagosome formation step. Just I want to show you the early step, how early step is occur within cell. Okay, well. Oh. Sorry. Wow. Okay. Now, okay. Just after this talk, I will show you the movie. Okay. Now we are working on the just a fundamental question I asked first. What is the what is, when and how cytosolic materials are degraded via autophagy. We need lots of works. What is induction condition, and what is a signal transduction for induction. And we want to know about the really target of the autophagy. For example, what kind of protein recruited to the vacuum autophagosome. It's not, I believe it's not simply bulk non-selective degradation. Some proteins are preferentially degraded by autophagy. And I think there are many, many modes of autophagy. Still, we should work this kind of question. Also, not so many people interested in degradation process and degradation product via autophagy. So we want to know about this process. And so without this kind of data, we cannot say really the physiological meaning of autophagy. One example I was shown, the autophagy was thought as a protein degradation system, but recently we found lots of RNA because you see lots of ribosomes is taken up into the vacuole via autophagy. So these RNA should be degraded, and we found the enzymatic process of RNA degradation within vacuole. T2 type nuclease is important, and 3' nucleotide is formed, and nucleotide come out from these vacuole, and nucleoside immediately degraded to the base and the ribose, and these bases are almost completely come out from the cell. So this is not just simply recycling, but nucleotide metabolism is very changed during this kind of autophagic degradation. So this is a paper of the autophagy-related research. I started the autophagy work here in 1988. At that time, only 20 or 10 papers per year, and not so many people interested in protein degradation, and autophagy was not popular in even cell biologists. And we found the autophagy in the East. First paper appeared here, and we found several interesting gene function of genes, but it, as you see here, now autophagy field is really become a quite popular field in cell biology, shown here. But I want to say it takes 10 or 20 years to become this kind of a hot field in biology. So I want to say many people, young people, I want them, you can work this kind of really popular field if you can really get you feel you will have a good result. But these are not a competitive field, but I think some challenging person start something somewhere here, and we make this kind of new field. So just 27 years of 29, almost 30 years of my autophagy research was really not so flat, but was helped by many accidents and wonderful occasions. And I really thank all the good collaborators and friends. And I want to emphasize my whole work was just a curiosity-driven research. I never thought the autophagy field with be related to the cancer or many kind disease. That's quite now popular field in autophagy research. But so I want to emphasize curiosity-driven research is very important to that. And this is a message for young generation from me. Okay, that's all.